At its very core, drug science must remain independent. This means we don't accept sponsorships. It's with the support of the drug science community we're able to do this and make the podcast in the first place. If you're able to become a drug science community member and support the show, you too will be supporting the dissemination of evidence-based drug policies. Without you, none of this would be possible. For anybody interested, there's a link in the show notes. Thank you. Hello and welcome to the Drug Science Podcast with me, David Nutt. Here we're bringing together experts and activists for a rational, honest and informed conversation about drugs. Well, hello and welcome to another Drug Science Podcast. My name is David Nutt, and tonight I have with me Deborah Mash, who is a pioneer of innovative research in addiction. And to me and many others, she's known as the lady who is taking Ibogaine from the field into the clinic. Welcome, Deborah.
Thank you.
So I'd like to, kick off these conversations by giving people a chance to tell us a little bit about their background. And I presume you're a pharmacologist by original training, were you? Or tell us a bit about how you got into science in the first case.
Yes, I identify myself as a neuroscientist, neuropharmacologist. I did an undergraduate degree in psychobiology back before neuroscience was a formal discipline. And from there, I advanced to do my PhD in pharmacology and began working in the field of Alzheimer's disease, working in the cholinergic nervous system. And after I completed my PhD, I spent a few years up at Harvard Medical School at Beth Israel Hospital in the impressive division of the late Norman Geschwin. Dr. Geschwin, sort of the pioneer, one of the most brilliant neurologists, behavioral neurologist, and working in the laboratory of Dr. Marcel Meslem.
And what were you doing there?
After that, I was doing cholinergic nervous system.
Was that Alzheimer's too?
Yes. So when I started, I actually was working in the cholinergic nervous system before they cloned the receptors. So we published, my dissertation was published in Science magazine, and that publication got me into Harvard, and the rest was kind of history. But my original NIH grants were all in the field of Alzheimer's disease. So I traveled back from Boston to Miami because at the time I was married to a young attorney, and he had passed the Florida bar, and the University of Miami Miller School of Medicine had made an offer to me to join the Department of Neurology. And I was a PhD in the department, and there weren't that many PhDs that were on tenure tracks, but I was invited to come in on a tenure track. I was actually the first woman hired on a tenure track in that department. And there I was, and I set up my laboratory and got my first NIH grants in place and was working diligently with several really impressive collaborators and working on nerve growth factor and again, continuing and looking at the cholinergic dysfunction in the disease. And at the time, things were changing up in Miami. So you recall Miami was on the front end loading of the cocaine epidemic. Because of our location and the transshipment of cocaine through the Bahamian corridor, Miami was hit hard. And we got hit not only with powder cocaine. Miami was the Miami Vice capital. So you couldn't go to a party, you couldn't go to a club, you couldn't go anywhere in Miami where cocaine would have come out. Restaurants and bathrooms, people were, hitting cocaine. And of course, with that came the deaths and the emergency room admissions and crack exposed infants. And Miami, we really felt it on the first wave. And one of the other things that I had done is that I'm a brain banker. I set up the University of Miami Brain Endowment Bank, which is one of the largest postmortem collections of human brains. Patients, people, family members who donated the brains of loved ones to support medical research. And at the time when cocaine was showing up in the medical, deaths were showing up in the medical examiner's office, I got looped into the addiction space because an old friend of mine from the Center for Disease Control had come down to Miami, and they were trying to figure out why people were dying with recreational blood levels of cocaine in blood. And someone said, well, you ought to look at the brains of these people. And the medical examiner said, well, we don't really have anyone interested in that. And one of my colleagues said, well, there's this woman, Deborah Mash, and she has a brain bank. And it turned out that my colleague from the CDC knew me from undergraduate days. And so the rest was history. And it was really that change up. I wrote my first NIDA grant, and had never published in the field of addiction at all. Addiction neurobiology was not my area. But I was so intrigued by these, what was the effect of cocaine on the brain and behavior, that I thought, well, this is very important. And we have a collaboration with the medical examiner's department, so we can have these brains donated for research. And at the same time, we were seeing cocaine psychosis, this very strange behavioral anomaly of cocaine-related excited delirium, which today is a very controversial topic in and of itself. But we had these cases. So, there we were in the middle of the cocaine epidemic, and I changed gears, and I write my first grant to NIDA, and I fund it on a first submission. And you and I both know that getting grants funded on a first submission is not a usual occurrence, but there I was. I had my grant.
Yeah, that was an emergency in a similar way to COVID's a bit of an emergency. That was such a tsunami of cocaine deaths and the rising use. I mean, it was pretty vital that people started to research it. So absolutely, you saw that was needed, and well done.
Well, thank you for that. I mean, we did. We really changed gears. And what was interesting is that we, in the first year of the grant, in the first year of funding, when we got the money, and I sat around the table and I assembled the team, and I said, all right, NIDA's given us this money. What exactly are we doing with this money? We need to address this problem, as you pointed out. This was an urgency, and we needed to kind of hit the deck running. So my colleague said, well, Deborah, you ought to look at coca-ethylene. And I said, coca what? Coca what? And he goes, it's the metabolite that's formed when you drink and use cocaine in combination. And I said, no, I don't know what that is. Draw it on the blackboard. So he sketched up the molecule, and I said, can you make it for me? And he said, yeah, I can synthesize some. And we took it to the test tube, and we presented at the Society for Neuroscience, and I was invited to do a press conference. We were the Miami Vice metabolite, cocaine and booze, lethal mix.
Yeah, actually, on that point, yes, some of our listeners won't be familiar with. To tell a little bit more about what it is. Then it's when cocaine and ethanol fuse to make a new molecule. Is that right?
Yeah, exactly, David. It's a transesterification. So cocaine and ethanol in the body will form this third molecule, and it's the ethyl homologue of cocaine. And what we were able to demonstrate, we were able to demonstrate in the test tube that it had a high affinity for the cocaine binding site in the brain, the dopamine transporter, but it also, if you administered it, you could demonstrate that it was more lethal. And so we went on from there, and we did some epidemiology studies, and we did some tox testing and, cadaver deaths. And we had this really nice story, and we go up to the Society for Neuroscience. They pick it for a press conference, and it was amazing because I was a new person at NIDA. No one knew really who I was. I was an unknown, and now here I am. And in the first year of funding, we're getting national recognition. And this is really what leads us to Ibogaine, this little experience of working on this epidemic, because we get national recognition. I go in there, and it was the old days when, you had slides, and I go into the press room.
Oh, yes.
And I put my little carousel on the thing, and I look at all the reporters, and I go, let me tell you a detective story, and everybody's pen goes to paper. And we were written up in Science magazine, again, as the Miami Vice metabolite. We were written up in JAMA. We hit the cover of, I'm not making this up, every newspaper in the land. People were contacting me for interviews. I mean, it was a ridiculous amount of press. But in the context of this and all this media, three things happened. I like to say good things happen in threes. And The first was that the president of the University of Miami, the late Tad Foote, invited me to speak at the Coalition for a Drug-Free America. You remember that in those days very well. That was the Just Say No to Drugs time.
That's not some Reagan, was it Bush? I can't remember which president's wife was driving it.
Yeah, that was Reagan.
Reagan.
And so there we were, and of course your university president asks you to speak, you go. So I go, and I give my talk about cocaine and the cocaine epidemic in Miami-Dade and everything that was going on with that. And this gentleman comes up. I was surrounded after my talk. Many people had come up to the podium to ask me questions. And there was a Black gentleman who walked up to me, and he was very animated, and he wanted to tell me about this drug from Africa that could be used to wean people off of heroin, cocaine, and other hard drugs. And I listened to him for a few minutes, and I had no idea what he was talking about. And I think I was rather abrupt with him. Like, he was so animated and really wanted me to understand what he was telling me, and I had never heard of it, and I thought it sounded far out. And so I was abrupt. But that was the first time I had heard about Ibogaine. The second time I heard about Ibogaine, I was presenting again. I was invited to give a lecture at the College of the Province of Drug Dependence. And I went in there and I gave my talk again on cocaine. And I was getting really sick of listening to myself talk about, cocaine and ethanol mix. Yeah. And I remember sitting in the back of the room. I was unprepared, and I had my program, and it was a session on cocaine. So I said, well, I'll go sit in the back of the room and look at my book. And I sit in the back, and this professor from Albany, Stan Glick, is up there, and he's feeding this substance from Africa to rats, and they stop drug-taking behavior. And I thought, oh, that's that same molecule. And the third thing that happened was when I came back from that meeting, and I had an old answering machine in my office, and I played the tape, and it was Howard Lutzoff, the late Howard Lutzoff, who made the seminal discovery of Ibogaine and reported on it, calling me because he wanted to use our work on coca-ethylene to support a polydrug dependency patent that he was filing on Ibogaine. Now I've heard it 3 times, and I said, Howard Lutzoff, you're the man behind Ibogaine? And he came down to Miami. I said, bring your data. I want to see, how does it work? What's the mechanism of action? What do you know about it? And he goes, okay, I'm going to come to Miami and I'm going to show you. So he flew into Miami, and he came in with, literally, David, a box of papers, and they were kind of junky papers, and they just newspaper clippings, and it wasn't data like you and I are used to looking at. But he did something. He invited me to go to Amsterdam and see it with my own eyes. And I did.
Oh, they were using it. They were using it in Amsterdam. I didn't realize that. It was being used as a therapy there, was it?
Yes. They were running an underground railroad of addicts helping addicts. And it was ICASH, the International Coalition of Addict Self-Help, and the Dutch Addict Self-Help Movement.
Uh-huh. I didn't know. So you went to Amsterdam. Right.
I did.
A lot going on there as well.
I brought a medical doctor with me, of course. And so a colleague, an MD PhD who was in my department, Dr. Juan Sanchez Ramos, I said, Juan, will you go with me to Amsterdam? I want to see this. And I bought tickets, and we flew over to Amsterdam, and we stayed in residence with them. And I saw 3 young men take Ibogaine and go through this transformative experience. And I couldn't believe it. I saw a young man who was on over 100 milligrams of methadone, chipping on heroin, coke, and benzos, take a single dose of Ibogaine, and in less than 30 hours, go to the bathroom, shower, shave, clean up, eat a big meal, and sit down across the table and talk to me about what was going to be different in his life. And another young man who was a musician, a very talented Puerto Rican gentleman from New York, who was banging back a lot of heroin, really hardcore use. And he had the same experience, no withdrawals, no desire to go walking out the door and getting high. And again, thinking about what he needed to do to change his life so he wouldn't fall back in the hole. And the third one was the son of an Orthodox rabbi from New York who had been freebasing cocaine. And he had the similar experience where his desire to go out and get high with coke was diminished, and profoundly so. And I couldn't, seeing is believing, right? But it was so, I will go to the grave with that experience, because that is what has fueled my interest in this ever since, is that what I wanted to know, why would a single oral dose of Ibogaine be able to reset all this wreckage and completely diminish bring about a gentle detoxification. No, none of the acute withdrawal symptoms, chills, diarrhea, vomiting, nausea, diarrhea, muscle cramps, all the anxiety, severe depression, the post-acute withdrawal symptoms, the anhedonia, all of this was just gone, diminished, not unseen. How could this be? And so I couldn't really do any research there, because although they had a medical doctor a psychiatrist who had worked with Holocaust survivors who was there, Dr. Bashan, there was no cardiac monitors. There were no IV poles. There were no blood pressure monitors. I mean, this was not the setting that, when I arrived there, I thought I had actually lost my mind, because this wasn't really a good medical setting. But I had an idea. I said, maybe there's an active metabolite. And I asked the young man, I said, can we collect urines? And so I did. So I got one of Lutzhoff's associates to get me dry ice and a styrofoam container and urine cups, and we collected urine, and I brought it back to Miami, and I gave it to my same.
You brought your souvenir from Amsterdam.
I brought it back.
Most people bring food. You bring urine.
I froze back a bunch of urine, and I put it in the box. I got it. I went to the airport there and at Schiphol, and I gave people money, and I said, make sure that box gets on the plane. And that box got to Miami. I got the box off the plane, drove right to the medical examiner's office, to the toxicology division, handed them the box and said, show me the metabolite. And that's how we identified Noribogaine.
So what made you think there might be a metabolite? I mean, was it a hunch?
David, I had just worked on a metabolite, cocaine.
Oh, I see. Aha, right. I've got it. So it was, yes, extrapolating from the cocaine. So you found this metabolite. That's some desmethyl Ibogaine. Is that what it is? Yes.
Yeah, we figured out pretty quickly that it is an active metabolite of Ibogaine. So whereas Ibogaine is cleared from the blood within less than 24 hours, Ibogaine is gone, cleared out of the blood. Noribogaine is elevated. So in the days after Ibogaine, Noribogaine is elevated in blood, and it lags behind the Ibogaine because it goes through the liver and is, it's a first pass metabolism. So we thought, well, maybe some of the beneficial after effects of Ibogaine are due to the active metabolite.
Yes, well, that's very sensible. Yeah, quite credible. So then you went off to, and that's quite a few years. You've been searching Noribogaine for a long time then now.
Both molecules for a very long time. So that was 1993. And I came back to the University of Miami, and I had a wonderful, my dean of research, Dr. Robert Rubin, incredible dean and mentor, and just a wonderful human being. So I came to him, and I had a good reputation at the medical school. I wasn't, I had a couple of home runs, and my funding was very good. So I went to the University of Miami, and I said, we have to test this. Ibogaine needs to be tested in an academic medical center. So, my naivete. And so we have to submit an academic investigator-initiated IND to the FDA, and we're going to do it. So I had to go up in front of the university, get permission. Then I had to go up in front of the insurance carriers. And of course, we have to pay for this. We have to find the money. And so I go, all right. So I go up to NIDA, and NIDA had invited me to give a talk on cocaine again. And so while I was up there, the head of the medication development division, Dr. Charles Krajinskis, and Frank Vacci, the deputy, I talked to them afterwards, and I said, University of Miami is going to submit to the FDA. We're going to submit an IND. And David, I swear to God, I think when I turned around and went to walk out of the room, that both of them laughed at me and thought, oh yeah, right. You're going to do that. Okay. But I did, undeterred.
Little did they know. Little did they know you.
So I submitted it and we went up before the Drug Abuse Advisory Committee. So these were the same people at the same time that Rick Doblin was going in with with his early work at MAPS. So there were few of us. I mean, we got permission from the FDA in 1993 to do the phase 1 study of Ibogaine. And at that time, of course, I was proposing that we would do it in cocaine-dependent patient volunteers. And again, the same group came at us. And we got permission, actually, we put our protocol in. The IND went up and our clinical trial protocol goes in. And my medical safety officer, Dr. Dan Spiker, 30 days they give you the call, right? So I'm sitting there at my desk, and day 30, we get the phone call from Dr. Spiker. And I ran and got Juan Sanchez Ramos, who was our clinician on the study, and we sat there on the phone with them, and they said, we're going to let you go. We're going to let you do it once you make a few changes, but you're good to go. And I hung up the phone, and I couldn't believe it. I said, oh, my God, they approved this? Wow. And so, okay, so now we're going to do it. And long story short, a few months later, we got a phone call from Dan, and he said, Deborah, have you put Ibogaine into humans? I said, Dr. Spiker, we're still, getting ready to start the trial. No. And he said, well, I need to tell you something. There is a group of investigators at Johns Hopkins, and they said that Ibogaine causes cerebellar toxicity. And that was George Riccardi's colleague, Mark Molliver.
Oh, right. Yes. Okay. I'm vaguely remembering that now.
Yeah, it was kind of the same strategy, raise the neurotoxicity flag, slow things down.
But Ibogaine's not a recreational drug. Ibogaine's a therapy.
Yeah, Ibogaine is not a recreational. No one is taking Ibogaine in a dance hall scene. But, it's used for the single purpose.
Well, that's right. But you highlight a very important problem, which is the groups who spend their lives finding problems with drugs, they find problems with drugs. Because I don't think the cerebellar damage turned out to be of any relevance, did it? Or was it meaningful?
No, but it did appear recently, flagged up again when Senator Schatz and Congresswoman Alexandria Olivia-Cortez raised the issue to the Congress and to the Senate about increasing funding for advancement and testing of psychedelic medicines. And it not only included psilocybin and MDMA, but they also mentioned Ibogaine. And probably from the press office at NIDA, I assume that that's just the group that wrote the reply back to the senator and the congresswoman, named this issue of neurotoxicity with Ibogaine. But, there has never been yes, there is cerebellar ataxia early after drug administration, of course. When you're at the height of the oniric effects of Ibogaine, you really can't walk around. You're down for the count at that time. But we did, I was in a neurology department, so we did full neurologic assessments pre and post. We did bedside posturography. We looked at tremor analysis. I had wonderful neurology colleagues, and we did all that. And we could demonstrate nothing, except a transient ataxia. So there was really nothing there. And I had also done some monkeys. I had been down in St. Kitts working at a primate colony on an NIAAA funded study, and I got my hands on some primates. So we did repeat dosing in the monkey, and we showed that at the therapeutic levels, there was no cerebellar toxicity, and that was independently reviewed by neuropathology colleagues at the medical school. And then last but not least is that there was a young woman who died from natural causes. Remember, I had the brain bank. So some of Luxon's people would come through Miami. And we would do pre and post evaluations on them. And when they would sit and we did MRI scans and whatnot, I would talk to the young people about, you should donate your brain, your iboganauts, and God forbid if something happens to you, your brain would be very important to be donated. And lo and behold, one did pass away, and her brain was donated, and I was there at the time of the autopsy. We brought the brain back, and we submitted that information to the FDA, and there was no toxicity. And she had taken Ibogaine actually more than one time with Howard, and she had used Ibogaine, detoxified, stayed clean and sober for a year, and then a year later she relapsed and she went back for Ibogaine. So she had more than one dose, and her brain had no toxicity.
So how much did it set you back, that scare, the cerebellum scare? A few years?
No, it slowed us down, David. Absolutely slowed us down. And in fact, I went into before I did an open hearing at the FDA. And the FDA, that was a mistake too. I thought everyone would want to know about this. I was so seeing is believing. I wanted to share this with everyone. But at the time, buprenorphine was just coming down the pipeline, and there was a move to repurpose buprenorphine, which is now a multi-billion-dollar industry in our country. And here I am with this, transformative oniric medicine that could take people to abstinence. Nobody really wanted to see this. I was I have to say, very naive. But anyway, the undeterred, I come from an aspirational blue collar family, so it takes a lot to knock me down. But we went up in front of the Drug Abuse Advisory Committee, and in the middle of the meeting, when Dr. Molliver hit me with his cerebellar toxicity, and he was showing angry astrocytes. This is one of the two cell types in the brain, and astrocytes can be a marker of neuroplasticity, but they can, of course, also be a marker of toxicity and degeneration. So he's showing this, and everyone is, clicking, and the newspaper reporters are writing up, and everybody, oh my God, the neurotoxicity. So I went over to Mark, and he had no quantitative data or anything. And I went to him, and I whispered in his ear in the break, and I said, Mark, are you sure about your data? Because people are dying from cocaine.
Yeah, quite.
Mark, please. And of course, he had a contract from NIDA, and, people have conflicting, as you know very well, and as I know very well, people have conflicting interests. But we got past it. So I put it into a closed door session. I'm not even sure I knew how to do that, but I did. I closed the session, and I went in there, and I came out with approval in 93 to test. And in 1995, I went back before the FDA again in a closed door session. FDA was always highly collaborative and helpful. Next time I went in, I had Charlie Grudzinskis sitting by my side. And actually a week ago, I was on the phone with Dr. Grudzinskis. He runs a private consulting group, and he's helping me today. So, never say never. You keep going, right?
Well, that's right. I mean, you're talking 30 years, but you're making considerable progress. So, tell us where we've gotten to now.
Yeah. So fast forward. So in 2017, in 2010, I actually, because I couldn't fund the Ibogaine study, so we had FDA permission, just the cliff notes on this. When we had FDA permission to go forward, FDA extremely collaborative and helpful, and they always have been, by the way. And, but I couldn't fund it. And Howard Lutzoff held the intellectual property, so I couldn't raise funds. And I wrote a NIDA grant, and NIDA didn't want to fund it. And then NIDA was going to put their own clinical program together around Ibogaine, and they had convened an external panel, and the panel split. So the industry group, some of the academics thought it should be tested, and the industry group thought Noribogaine should be tested. So I went offshore. I went ex-US, and I never intended to do that. And the University of Miami, amazing academic institution, and my chairman and others at the school allowed me to do this. So we went ex-US to collect the data because I couldn't give it up. But I didn't want to run a clinic ex-US. I wanted to be back in the US. So in 2010, I started a company, and we thought we would take Noribogaine forward and we would leave Ibogaine behind. Fast forward in 2017, at the height of the heroin epidemic, when the deaths were rising, I couldn't take it anymore. And I said, you know what? I need to finish what I've started. And so I decided to leave the university and to go in and take over my company that I had founded. And I went in and did a reorganization in Demorex and decided that we would take Ibogaine, and we'd have two jockeys, two horses in the race, and we'd take both drug products in, and we would bring back Ibogaine. And that's what I did. And the fortunate thing is that I was able to find an investor. Howard Lutzoff had since died, and we had some new IP pending around Ibogaine and some position. And And we were able to forge a joint venture with Atai Life Sciences. And then the question was, where do you do the clinical work? And, for me, I'm in the US of A, where Americans consume more drugs than any group in the world, as we all know on this call. And I wanted to, bring this home. I wanted to bring this back to the US. But my colleagues at Atai decided that they wanted to go ex-US, collect seminal data on the safety of the drug, and really put some of the concerns around Ibogaine to rest. And that's, of course, an important strategy because people have taken Ibogaine. When I closed down St. Kitts, there wasn't a place where people could go and get Ibogaine safe. And so what has happened is it pushed Ibogaine into this vast underground where you have pop-up clinics and people buying internet Ibogaine and got, the purity unknown. And people administering Ibogaine who are not medical doctors. Opioid detoxification is a medical condition. You need to be under the care of a physician, a doctor, someone who understands the metabolic disruption that has occurred in your body. And people who abuse hard drugs damage their hearts, their livers. I mean, these are not healthy people. So, and then there's the psych comorbidities. So you really need to know what you're doing here. And of course, there's a lot of unethical people who sell Ibogaine. And so people have died. And we at Demorex, with my colleague, my chief medical officer, Dr. Matthias Luz, have reviewed all the Ibogaine-related deaths, and there are 33 of them. And we've determined, based on our review and trying to give a causality assessment, that the reason people die is precisely what I said: unsafe settings, taking, toxic doses of the drug, having, disruptions in their magnesium and their potassium and their blood levels and their enzymes and things. It sets them up for toxicity. So that's a big hurdle, and we need to beat that back. The other thing is that there is, Ibogaine acts on the heart, and there's hERG channels, which are potassium channels in the heart, and Ibogaine in certain concentration will bind to those hERG channels. So will noribogaine. So we're learning a lot about the therapeutic to toxic window. And we know, unlike other psychedelic medicines like psilocybin, for example, which has a very wide therapeutic index, Ibogaine is going to be given in a more narrow therapeutic index. So it has to be done in a well-controlled medical environment by people who are trained and understand how to work with this drug. So that's what we're doing now. And the fortunate thing is our clinical trial is starting up. We have permission from MHRA in the UK to test Ibogaine in a good clinical trial sanctioned study. The protocol has been reviewed by the MHRA, by the regulators in the UK, and with their guidance and support and the support of the ethics review panels, we're able to get started. And so we're very excited about this. We're enrolling now, and we're looking for volunteers in the first part of the study, which is going to be healthy volunteers who have had a past experience with a hallucinogen. And they'll be part of the study to help us learn more about that safety window, and then also to rule out any cardiovascular risk.
Yeah, I think I tweeted your advert the other week. So I hope that had some impact on your recruitment.
Thank you. With COVID, David, it's been slowly moving. Our recruitment has been slow as people are coming out of lockdown and don't really want to go, right back into a study. So yes, we do need to get the word out. We need volunteers to help us advance this transformative therapy.
Actually, and that's an interesting point you raised. So people listening to this, if they want to volunteer, how best to access that? Maybe you can give me a link. We can put it up on the website. And then if people want to volunteer for your trial, they can go to the link. So we'll do that at the end.
Yes, I appreciate that. We'll give the link so that people can find out more about this. But the site is MAC, capital M, capital A, capital C, in Manchester, UK. And that is the early phase unit at MAC Manchester.
Super. So you've achieved your goal. You're going into what you might call, the first stages of development of Ibogaine as a medicine. But let's just roll back a bit, because there are two really interesting aspects of Oh, Ibogaine. I mean, the first, I suppose, is the pharmacology. Do we know anything more about it? I mean, it's still a bit of a mystery, isn't it? Because it's not a classical psychedelic, I don't think.
Not at all. I had a wonderful experience in that, when I was starting out, I got invited to go to Esalen, back when Rick and some of the others were known people at Esalen, but nobody knew Deborah Mash. But I got invited, and I met Sasha Shulgin.
Oh, right.
Dr. Shulgin. And Dr. Shulgin, I asked him the same question, David. I said, what do we know about this molecule? Let's talk about the chemistry. Again, as a pharmacologist, as someone interested in looking for druggable targets, I wanted to know all about it too. So we sat down and he drew 4 boxes on a piece of paper and he said, Deborah, LSD and psilocybin in one, the tryptamines in the second box, the hallucinogenic amphetamines, MDMA, in the next box, and Ibogaine in a box by itself. And that's what he taught me. And well, in answer to your question, so Ibogaine acts on serotonin receptors, weak agonism, and it also acts on NMDA, right? So it's ketamine-like. So it's serotonin and glutamate on the Ibogaine side. Then it gets converted to noribogaine, and noribogaine has a very interesting pharmacology. So noribogaine is a serotonin reuptake inhibitor, but an atypical serotonin reuptake inhibitor. It interacts with the transporter in a way distinct from other classical serotonin reuptake inhibitors that are approved drug products. Ibogaine also acts on the site, but with much less potency. So the affinity constants are lower, and it's less potent at the site. So noribogaine is very potent on serotonin reuptake. And then it also is a partial kappa agonist. So it is acting, so it's kind of like, I call it a kapatonin, kappa and serotonin. So Ibogaine is acting on serotonin and also the glutamate pathway. And that's probably the visions. Visionary experience. Because we know when you give noribogaine as the sole agent, you don't have the hallucinations. You don't have the waking dream state that patients report when they take Ibogaine.
Oh, so the noribogaine has taken out the glutamate and replaced it with some capergic effects.
There's no glutamate affinity. The dwell time in the glutamate channel is negligible. We published that a long time ago. So there's no glutamate effect, but it's kappa. And then the little, and to what extent this really fits in, we don't know yet. But there's also, Stan Glick did this work in looking at the Ibogaine conjugator 18-methoxycoronardine, where he demonstrated an effect on the alpha-3 beta-4 neuronal nicotinic receptor. So that site is interesting because that's the habenula. And the habenula feeds back into the dopamine cell body fields. And you can kind of think of it as putting the brakes on dopamine-mediated reinforcement. But there's also some growth factor effects. So there was an interesting set of papers put out by Dorit Ron, and she's at UCSF, University of California, San Francisco, at the Gallo Center. And she published a seminal paper looking at noribogaine and not 18-MC. And she'd also published on Ibogaine also, and demonstrated that glial-derived neurotrophic factor, GDNF, which we know is important for mediating the reset on dopamine neurons, is mediated through an action of noribogaine. So now we've got a perfect thing. When I think about addiction, obviously, when you think about withdrawal and what the circuitry is in withdrawal and why you've got all these, symptoms and protracted symptoms when you come off of drugs and alcohol, and all of the monoamine-containing pathways express mu-opioid receptors, but some of them are dopamine, some of them are serotonin. So you have this neural circuit, and it seems to me that Ibogaine and its active metabolite is uniquely hitting a cluster of targets, and it's this polypharmacy effect that's bringing about this rapid reset.
That's very interesting. So I've often wondered whether Ibogaine got a bad rap because it was being used in withdrawal. which is obviously the most dangerous time because people can die in withdrawal. The cardiac effects, particularly of things like heroin and withdrawal, can be profound. Do you think it has to be given in withdrawal? Do you think it might work if people have been withdrawn in another way and then could go into an Ibogaine session where there's less risk? What do you think in terms of how this might be rolled out clinically?
I think that's an excellent idea that, Ibogaine could be given for post-withdrawal management. However, if you think about, David, how are we handling detoxification today? How do we do that? How do we, get people off of drugs? And you even think about giving them an antagonist like naltrexone, which is beneficial post-detoxification. If you try to taper right now, the only approved molecules for this, the only approved drugs are two of them. You've got methadone taper, which is approved. Buprenorphine is used off-label as a taper. And then clonidine is used, that alpha-adrenergic clonidine is used off-label. And lofexidine was approved in 2018 for withdrawal management, non-opioid. That's it. And so many people do not complete successful detox.
Yeah, I can see what you're saying. Yeah.
This is a problem.
Yeah.
This is a problem. So, it'd be interesting. Your point is very good. You could give a lower dose, for example. You could imagine giving a lower dose of Ibogaine in the setting of detoxification, much like brexanolone is used under full medical monitor for a postpartum depression. The new molecule Zulresso that came out of NIMH for the GABA reset. So you kind of think of Ibogaine as the glutamate serotonin reset, and then as compared to brexanolone or Zulresso. So you do that under medical, full medical monitor, inpatient setting, and you could give a lower dose of Ibogaine, very good, and then let them start on their, road to recovery, and then come back in 30 days when things are stabilized and do a repeat dose, higher dose where you use it in a therapeutic way for psychotherapy, in other words.
Yeah, absolutely. Because the problem, let's not beat about the bush. The problem with Ibogaine have been those deaths. And there are many, many people who are absolutely terrified of it. And some people would say, well, Deborah, you've taken on an insurmountable burden because you're carrying that legacy and you're trying to get this drug, over that terrible historical burden and into the clinic, and anything you could do to minimize.
No, David, I think about this absolutely. I think about this all the time. And all I can tell you is, we had 277 Ibogaine patients go through St. Kitts. We had not one death, not one hospitalization, not one arrhythmia. And believe me, when you go back and we had external people go through the charts and review our data, really looking at this, because we needed to have that kind of independent review of those charts. And originally, when I started out, we had Holter monitors on the patients. I was worried about this. I mean, lots off had had a death, and we were keenly aware of this. So I do believe that Ibogaine can be given safely. I do believe that, and I believe the data is there, the clinical data is there to support it. You have to know what you're doing, and you have to have doctors who can manage things. I mean, there are protocols to manage. We've done it, and we know how to do it, and we can train others to do it. So I think that's going to be the hurdle for us, is to demonstrate that to the regulators. And is Ibogaine, other psychedelics are being used for, therapy, right? So many people who abuse drugs and alcohol are self-medicating, as you as a psychiatrist know this very well and better than me, other comorbidities like major depressive disorder, trauma, they're suffering from. I had a wonderful conversation with Dr. Rachel Yehuda recently, and, she talked about trauma and addiction. And oh yes, I mean, people are self-medicating those inner wounds. And so I think that we will get past this hurdle. We will demonstrate that Ibogaine can be given safely in a medical setting. And then the ability to, even think about this, if you detoxify with Ibogaine, and it is an adjunct to psychotherapy, there's no doubt about it. When I was telling my counselors, we had addictionologists and therapists with us in St. Kitts, and psychiatrists, and the whole gamut, practitioners. And I said, okay, the pharmacologist walks in, we're not giving Ibogaine. We're going to develop Noribogaine. And they knew Noribogaine didn't have the oneric effects of the Ibogaine. And they said, no, Dr. Mash, you can't do that. And, David, I tell this story, I say pre-contemplative, contemplative, ready for change. That was the thing about Ibogaine. It was so dramatic. Not only was the withdrawal management dramatic, and that it was a gentle detox, and that we were able to, deliver people back into the treatment, to the little treatment community and let them work with their counselors, the psychiatrists, and the therapists. But the fact was that they were intractable users. These were people who came to St. Kitts who failed the standard of care. They had tried detox many times.
Yeah.
Many times. And they were just able to, the clarity of, I've got to change it up. I've got to change my life now.
Yeah. It's very reminiscent of what we see in our depression studies, where people often report that, psychedelics reset the brain. It's like they defrag, or they, reformat the computer. You go back to your base state, and so you've sort of wiped the slate clean. And it is very compelling. And it's also, I think it's kind of credible, given that we now know that disorders like addiction and depression, that they're system disorders, they're disorganizations of complex brain networks. And to expect perhaps just a single receptor targeting drug to completely disrupt something as massive and as overlearned as addiction may be too naive. And, this new model that you actually disrupt the whole process, I think is very exciting. And, well, I wish you well, because of
transformative neuroscience, the area that you have pioneered.
Well, I was going to say, you've been going for longer than us, but it would be really looking forward to working with you to look at those brain images under Ibogaine and see whether we can look at the disruption of those pathways and maybe even image recovery. That would be such a lovely thing to do.
Well, we are definitely going to do that together, and the collaboration with you and your team, you've contributed some of, the most important findings to the field, and to have you look at, to study the EEGs, study the images of the brain, and to be able to help us identify that circuitry. I mean, I've lived for this, to be able to image. If Ibogaine can give us a window onto the brain and to the addiction circuitry, and how to heal that and reset those loops, reboot that computer. That's fundamental neuroscience at its best.
Undoubtedly. Well, Deborah, thank you very much for joining me in this podcast. And I have to say, hearing your story, it is remarkable. And you've got an enormous will and enormous resolution. And if anyone's going to bring Ibogaine to the clinic, it's going to be you. So I wish you well. I'm sure all the listeners wish you well. And when you get the license, come back on and we'll celebrate. All right? Thank you so much for joining me.
All right, David. Thank you so much. And thank you to your listeners.
Well, that's the end of this episode of the Drug Science Podcast. Thank you for listening. But before you go, I would just like to share with you a question from our Drug Science community members. Recently, we recorded a very special podcast episode in which we invited all of our premium and philanthropic community members to ask me anything they like. Their questions were so good, I thought we should include one or two of them at the end of every podcast episode. So please enjoy this new segment of the show. Apologies for the audio quality, as we recorded the session over Zoom. Hopefully they're vaguely relevant to what we've been discussing. And if you want to ask me anything, perhaps we could do an Ask David Anything Part 2. Enjoy.
Another question from Peter. And Peter would like to know that there seems to be a bit of citizen science going on at the moment, microdosing, and he thinks it's similar to the early days of CBD. And do you see microdosing psychedelics going the same way as CBD? So somewhat of a free-for-all with lots of different products available, like tinctures, vapes, etc.? Or do you think microdosing will be a much more regulated market with dispensaries, practitioners, or maybe even stay illicit?
Yeah, that's a really interesting question. Yeah, so I'm always a bit embarrassed to talk to people in microdose because everything we've done shows it doesn't work. But that's not entirely true. So we wrote this huge review a couple of years ago trying to look at the evidence, and we couldn't really find any evidence then. And then we did this very big study for microdosing, but of LSD microdosing, mostly LSD microdosing, citizen science, self-blinding, and it showed microdosing works. But so does placebo, if you think placebo is microdose. So the effect is largely a placebo effect. That doesn't mean it doesn't work, of course. But it does mean you couldn't license it, because there's a lot of resistance to licensing placebo. And we're not sure if you license placebo would actually work. So what's going to happen to microdosing? The answer is, I don't know. I think because you've got to do it daily or 2 or 3 times a week, the clinical trials are way more complicated, strangely, than doing the trip-based therapy, because trip-based therapy is just, for Compass, it's just one trip, and then everything else is back to normal. Whereas microdosing, got to come in and have it administered 3 times a week and and be around in a safe setting just in case something weird happens, because it's a very dangerous drug, so to be. So I think it's 4 years ago now, we got permission to do LSD microdosing, but they insisted every single dose had to be given in hospital. And for 6 weeks, for 3 times a week for 6 weeks, we couldn't afford the hospital beds, so we never did it. So I think it won't be like microdosing CBD, because CBD is legal. If psychedelics became decriminalized, then I think there will be probably quite a lot of black market microdosing. But whether, I don't know. I have a fear that psychedelic microdosing may never ever be easily available, unless we actually completely reform all of the drug laws and have these drugs available in pharmacies, which I would support, but that's kind of a few steps beyond where we are at present.